love Argonaut. I kid you not. There's so much to know about AGO. So, yeah, if you've been following me along, you're probably getting tired of me talking about Argonaut. But it's the protein I study, and it's really amazing, and it helps regulate the levels of like thousands of messenger RNAs, so protein recipe copies throughout the cells. So then in those copies, it can like reduce the levels of them in a sequence specific way, and so then less of those certain proteins are made. So it's this really, really awesome protein. And I want to today kind of like put together some of the concepts I've been talking about, so I'll tell you more about like the protein in like graphics form and like what it does, and then also what it looks like. So kind of show you an example of structural biology, so where you're connecting form and function and learning about one from the other. And so it's really cool because a lot of the work that I'm going to show you, so I can't show you like my unpublished stuff. Hopefully in the not too distant future, I'll be able to tell you guys more about that. But a lot of what we know about Argonaut was actually worked out in this lab. So I'm in like the Lemore Joshua Torres lab at Cold Spring Harbor Laboratory. And they were actually like the one that discovered that AGO2 was a slicer in RISC. Like seriously, it's an awesome place. Okay, but anyway, so I want to tell you a little more about Argonaut. So I'll start with that, just like I'm practicing for like a talk, and so I just kind of modified the intro to make it a little more intro-y. And I'll tell you a little more about structural biology and where these structures come from, and then how the structures help tell us about how AGO functions, and like how it moves in different ways throughout its functioning. And so yeah, so basically I've just been playing around a lot with making some graphics and slides and stuff for presentations. And that sort of thing. So some of them are still really rough. It's all a work in progress. And there's some like different representations and some jerky stuff and things. But hopefully it's still helpful for people, or at least it gives you a sense of how cool Argonaut is, because it's really cool. So let's take a look. Argonaut, or AGO as its friends like to call it, is the effector protein at the heart of RNA interference, or RNAi. So by effector protein, I mean it's the one that actually does stuff and gets things done. So what's it do? It helps regulate the levels of thousands of messenger RNAs and their corresponding proteins throughout the body. It's a pretty important role. And the really cool thing is it does it in a sequence-specific manner. By binding to these small RNAs, which can be a microRNA or an siRNA, to form the core RNA-induced silencing complex, or RISC. These small RNAs are about 22 nucleotides long, and at the 5' end is a 6 to 8 nucleotide long seed sequence that allows AGO to use these small RNAs as guides to find and bind to messenger RNA targets that contains complementarity to that seed. These sites are typically located in the 3' UTR, and they often have additional pairing beyond the seed, especially centered around the 3' supplemental region, nucleotides 13 through 16, for reasons that we'll see in a bit. After binding, AGO is able to alone, or with the help of cofactors, induce mRNA degradation and target repression. So we can do this in a couple ways, depending on the amount of complementarity in the AGO. So humans have 4 Argonaut proteins, and our main Argonaut protein, AGO2, is able to slice fully complementary targets. So it's able to actually cut them, and this exposes raw ends for exonucleases to chew the RNA away. In the case of only partial complementarity, as is the case with most animal microRNAs, AGO recruits cofactors, so it binds to the scaffolding protein of the GW182 family and brings in decapping and deadenylation complexes. Those remove the protective ends on the mRNA and initiate traditional mRNA decay pathways. All of this leads to less protein being made, and then once AGO releases the target, it can do this again and again because the core RISC complex, so that AGO bound to guide, is incredibly stable. It has a half-life of weeks, which means it takes weeks for like half of it to fall off, and then half of that to fall off. It takes more weeks, and so basically it's super, super stable. So the single RISC, so an AGO bind to a guide is incredibly powerful. And as such, Argonaut proteins are evolutionarily conserved and important for maintaining homeostasis, so maintaining correct levels of proteins and mRNAs in your body. And they can also be used to like regulate developmental processes. Because this is sequence specific, it makes it really powerful and valuable therapeutic and experimental tool. So there are actually therapies that use small RNAs to lower the levels of dysfunctional or dysregulated proteins. And in the lab, it's used frequently to knock down certain genes, so basically reduce the protein production. And it does this in a non-permanent way. So it's not gene editing, it's just reducing the levels of the mRNA copies of the gene, so like the protein recipes. And so it's this really cool process and this really cool protein at the heart of it. And so we know a lot about it in part by biochemistry stuff, so like doing experiments to try to like see if it binds things and that sort of thing. We also know a lot about it from structures. And so this is a really good example of how structural biology is informative. So structural biology is basically it's a discipline that explores the interplay between biomolecular form and function. So what a molecule looks like and how what it looks like is related to what it does. And so you can think of like a spoon versus a fork versus a knife, or the different parts of a Swiss Army knife. So knowing what something looks like can help you understand how it works and vice versa. And the way that we can find out what proteins look like, there's several different ways, including X-ray crystallography, cryo-EM, and NMR. And so I've talked about crystallography and cryo-EM in past videos, not really NMR, but I've never done that. Well, I've never done cryo-EM either, but everyone else in my lab has, and so I hear about it a lot. But these ones are done, the structures I'm going to show you of Argonaut were done with X-ray crystallography. Some of them were actually done in our lab years back by Elad El-Kayem, who was a scientist in the lab who was a great mentor to me and helped me a lot. So thanks to Elad. But anyway, going back to, so X-ray crystallography, I've done posts on that if you want to learn more about it, but basically it's this technique where we get a protein to crystallize, so we get all the protein copies to like stay stable in this lattice, so like it's kind of like a brick wall, but the bricks are proteins. Not that simple, but the proteins are all in the same order and they're like frozen in space, and then we shoot X-rays at them, and these really high energy waves, and then they interact with the atoms, so like the carbons and hydrogens and stuff, the electrons, so it's too technical to get into here, sorry, but basically they hit the protein molecules and then they get scattered by those molecules, and then the scattered waves interact with each other, and some of them cancel out, some of them add together, and the ones that add together make this pattern of spots called the diffraction pattern, and then you work backwards from that to get this electron density map, which shows you where the electrons were, and you get this blocky meshy thing, and then you build the atomic model into the map. And so I'm going to be showing you the atomic model. So that's where you like the scientists who solve these structures, they built this model into that map of things. So basically you have that mesh, and then it's kind of like finding the center of the mesh, so like putting the structure into that mesh. And I've talked more about crystallography in the past, but the key thing is that once they solve these structures, they deposit them in this thing called the Protein Data Bank. And it's a multi-continent consortium, and all of these structures are freely available on the web, and they're given these 4-letter codes. And so the first structure I'm going to show you is human Argonaut 2, bound to a microRNA called miR20, and this is the one that was solved by Elad in our lab in 2012. But you can search for like any protein here and find this and download the structure. You can also play with it on the web. So I've done posts on videos on PDB as well if you want to learn more. But that was just your introduction to where the heck these structures I'm going to show you come from. Because they take a lot of work, even though they just look like pretty pictures. So first let's introduce you to Argonaut. So Argonaut, on the top I'm showing you like a domain map. So this is basically a domain is like a part of a protein that like if you took it away from the rest of the protein it would fold by itself. So that's kind of like a structural domain, and there's also like functional domains. So like parts that do certain things. So sometimes proteins have domains, like different proteins have the same domains. Well, they're not really the same, but they have the same name because they're really similar. And sometimes they were made by gene duplication and that sort of thing. So like different proteins also have a PAS domain. And so I'm going to show you these one by a time. So we're starting at the N terminus, which is called N. This domain is called N because it's at the N terminus, so the amino end. So this is like the the starting end of the protein. And then you have the PAS domain, which is I'm shown in red, the mid domain, and the PW domain. Then you have a couple linker regions, L1 and L2. And so my boss, my PI, the head of our lab, Limor Joshua Tour, she likes to compare Ego to a duck. So if you look at it like a duck, you have the PAS domain being the head of the duck. And it's biting down on the RNA. So there's this the miR20 guide, and so the 5' end, so like the starting end of the RNA, is held in a pocket created by the mid and PW domains, and then the 3' end is bitten down by the big duck's beak and the PAS domain. So let's look in a little more detail. So one thing you can do, so when I was talking to you the last couple days, I've been talking about PyMol, and you can actually represent the protein in different ways, because remember what we're looking at is a model, and so you can draw the model in different ways to highlight different things and to show in different representations. So before I was showing you the RNA was just sticks, and now you can see that I'm actually showing you like the RNA bases. And let's take a look. So the reason why this, I said that complex was really stable. Well, this 5' end is like anchored in there. That phosphate group forms all these bonds in that pocket that are really stable, and then the RNA channels through the protein. So it might look like you might say channel. Well, I mean, right now it looks pretty open, right? But if we show the surface representation, so now we're showing like space filling. So the space that those molecules actually take up, because it's not just sticky things, like they're more bulky than that. So right now you're showing the surface, so you can't see like what's inside. If I put it on like transparent or like partial transparency, you can see both at once, but that can get kind of confusing. And right now my point is to show you that this actually like this channel that the RNA is traveling through. So right now we're looking at the PAS domain, and you can see there's this channel, and peeking out of that channel is the seed region. So that's the part that's like most important for targeting. And so the seed region is prearranged into this helix that can then sample, and then this region opens up when it binds to a target. And you can see here you have that supplemental chamber. So when I said there was like 13 nucleotides, 13 to 16 was a spot that, yeah, sorry about the end of the video, but when I said that 13 to 16 was a spot that was important. So let's watch that again. Okay, so we're at the 5' end. You can see the seed region. Zoom in on it, and you can see it's prearranged. So the rest of the RNA is pretty disordered, but this region is really prearranged. So basically it can like hop around along targets to till it finds the right one, and then it can open up once the seed binds, and it opens up this like supplemental chamber where it can do that 3' supplemental pairing. So in order to do that, a couple changes have to occur. And so if you look here, you have that yellow thing, so that's like called helix 7, and it's actually moving out of the way so that the supplemental chamber can open, and with the help of like the L1 linker region. So you have it moving up and away, and so you can see that the bottom part of the AGO, so like the mid and PW domain, stays pretty stable, but then the linker regions help move the PAS and end domains away from the rest of the protein. So it's really cool. So the reason why um, so there's actually, like in the seed, the first few nucleotides, so the first few letters matter the most, because they're actually, this helix 7 is actually forming like a kink, so only the first few letters are really like prearranged and sticking out. Then so helix 7, so like there's that like pre-seed region, it kind of like samples out different targets, and if those bind, then the helix 7 can move away, and this allows the region to start opening up, and once it opens up, then you can have that second nucleation, so that second like place where the double bind, where the duplex starts forming. And so yeah, so that's just an example of how we can, structural biology can be really cool, and most importantly, how ego can be really cool. And so thanks for following along.